Can you see it clearly?
Yes. Hello everyone. Welcome to Creative Biolabs live webinar series. To start, I would like to introduce Dr. Sarah Voglan to present our service and the products first.
Thank you, Kathy, for the introduction. Yes, welcome everybody, and thank you for attending our webinar. I would like to start with a brief introduction indeed of our platform and services. And I would like to take this opportunity to introduce our new Organ-on-Chip platform, as you can see here on the slide. So as most of you are aware, the drug development process is quite challenging, and it takes a long time and also high cost to develop drugs. And there's still a high failure rate, more than 90%. Mostly, well, there are several different factors contributing to this, but part of it is the limited predictive value that animal models have for human biology. And our Organs-on-Chip platform offers a solution for this and allows you to test your molecules in a human-relevant system, including patient-derived material and preserving tumor heterogeneity. We also provide control environments, including gradients, flow, and immune interactions. We have different options in our platform. We offer a single organ on chip, multiple organ on chip, as well as barrier tissue and disease on chip, and further customized tissue on chip models. We usually start with the design of the chips, or can assist you with the design. And then we perform the 3D culture and can monitor these cultures in real time, after which we perform the data analysis, and every step of this workflow can be further customized. So furthermore, going a little bit deeper into the different models that we provide, we have both human normal tissue organoids as well as tumor organoids for different systems in the human body. So we have, for example, intestinal systems, we have pancreatic or gastric epithelium, we have renal tubules, bile ducts, lung airway, and epidermis, just to name a few. And also most of those we have for tumor organoids, and also there we have other things such as reproductive system options. These can all be used in drug screening and evaluation protocols, as well as toxicological evaluation. And further, we can provide custom organoid modeling. And as an example, I would like to show you here our platform in action, which first, as the study design, we used a patient-derived colorectal cancer PDO model, and we evaluated a drug candidate here called CXR102 versus a control. And we found that this drug candidate is able to reduce the tumor volume, and we were able to replicate these findings then later on in vivo, which actually demonstrates this platform's predictive power for clinical outcomes. So these we can provide additional data upon request, of course, if you're interested. And in addition to this in vivo model, I would like to highlight that our tumoroid models provide many advantages. So they can mimic the in vivo tumor structure as well as heterogeneity better. They have a high success rate and rapid turnaround, and they're compatible with high throughput testing. And this allows to preserve the patient-specific heterogeneity as well as the different cell types that are present in these tumors, including, for example, fibroblasts, endothelia, adipocytes, or immune cells. And therefore, they can be used to support disease modeling as well as immunotherapy and resistance studies. So overall, these tumor models can provide advanced insights in the tumor biology. So we can perform, first of all, as I mentioned earlier, the chip modeling, then customized assays, as well as the downstream omics experiments, and then analyze all these data to provide a comprehensive report with detailed insights into your molecule of interest. So for comprehensive data readouts, I want to summarize that we have high content imaging as well as quantitative assays available, as well as flow cytometry and multiplex immunoassays for secreted factors. For the multi-omics integration, we provide proteomics as well as RNA-seq, single-cell RNA-seq, as well as the correlation of the molecular data with phenotypic outcomes. And for the drug development, so this can all be used to understand the drugs better, for example, to understand the mechanism of action and resistance, as well as compare the efficacy across compound libraries and support biomarker discovery and target validation. So overall, we aim to provide a comprehensive solution for 3D biology. Beside our services, we can also provide several products, including ready-to-use organoids, 3D culture reagents, as well as several cell lines and biospecimens that are relevant to your project. And with that, I would like to hand the floor back to Kathy for introduction of our esteemed speaker for today.
Yes, thanks, Sarah. And now let me introduce our speaker for today, Dr. Mohsen Akabari. He is Associate Professor of Mechanical Engineering, the Director of Laboratory for Innovations in Microengineering at the University of Victoria, an Affiliated Associate Professor at the School of Biomedical Engineering at the University of British Columbia, Adjunct Professor at Tarosaki Institute for Biomedical Innovation and Department of Bioengineering at University of California, Riverside. His research findings have been published in over 140 peer-reviewed journal papers and book chapters with an H index of 52 and over 12,000 citations. His work has been featured on the cover of 12 journals and reported in over 70 media outlets such as CBC News, Time Columnist, BBC News, Google News, and Science Daily. And before Dr. Akabari begins his presentation, I want to quickly remind our audience that we will have a Q&A session at the end of the presentation. And if you have any question during the presentation, please type your question into the Q&A panel, and Dr. Akabari will get to them after his talk is finished. And now let's welcome Dr. Mohsen Akabari. And Dr. Akabari, thank you for presenting your important findings to us today. And you may begin when you are ready.
Thank you very much, Creative Biolabs team, first for inviting me, and then secondly for being so organized and helping me to prepare for this presentation. And it was well done. Kudos. So can you hear me well? Audience,
yes, we can hear you.
From the audience, do we have any voice issues? Everyone can hear. No one, everyone. I can't see the chat box for now, but I just want to make sure that everyone can hear me well, and you can see my screen well, right?
Yes.
All good. Awesome. Great. So thank you again. I'm very excited and pleased to present and discuss a platform we developed over the past few years in my lab. We call it Tumoroid-on-a-Chip. And it has been used for modeling tumors, specifically for applications in immunotherapy and chemotherapy and also radiotherapy. But the focus of my presentation today will be on how this platform can be used for immunotherapy application, with the specific focus on glioblastoma, which is a primary brain cancer. Excuse me. What motivates us to do research in this space, and Sarah also mentioned in her presentation, is the challenges associated with the drug development process. This process is lengthy. It can take up to more than 10 years. It's very expensive. It can cost more than $2 billion, and it has a high failure rate. Up to 90% of the drugs that reach the clinical trial, they fail. And the primary reason for the drug failure at the clinical trial are safety and efficacy issues, which means that the process of drug development, starting from drug discovery here, followed by preclinical in vivo studies, they are not efficient. The choice of drugs that reach the clinical trial are not the most optimized one in terms of safety and efficacy. There's also, even after the drug passes the FDA review and is being approved, there are also challenges related to long time toxicity, off-target specific toxicity, with good examples of two osteoarthritis drugs that were developed by major pharmaceutical companies that had to be withdrawn from the market due to cardiac toxicity issues that were observed. And then it was followed by a couple of multi-billion-dollar lawsuits. And the cost for Drug failure is reported to be 65 cents for every $1 being spent on the drug development process. And then this cost can be translated into $50 to $60 billion in loss only in oncology clinical trials. So the numbers you see here show the magnitude of the problem. And we think, I think my team, and then I believe Creative Biolabs team as well, we think that the challenge is in the models that are being used for developing the drugs, specifically the traditional in vitro models, which are basically 2D monolayers of cells being grown on flat surfaces, which are mostly plastic surfaces, perhaps coated with proteins, but still the cells experience the hard surface, which is not similar to what cells typically experience in the body in most organs. These models, they cannot recapitulate the complexities of the organs and tissues in our body in terms of the presence of diffusion, in terms of the presence of tissue barriers, for example, blood-brain barrier, and then also in terms of organ-organ interaction. So animal models are, of course, more complex organisms. They have multiple organs. They're living organisms, and then they are good candidates for pharmacokinetic and pharmacodynamic studies, indeed, but they're costly. And there are challenges with interspecies variability. There are issues related to ethics. And then if you want to use them for screening applications, because they're low throughput, the use of these animals for these applications are not practical. Sarah mentioned, and I want to echo what she said, is that organs on a chip, in general, they offer benefits over traditional 2D culture systems. These platforms integrate 3D tissue architecture with fluid flow, and then also the ability to co-culture multiple cells that you typically see in the body in a three-dimensional environment. All of these enable more accurate testing of therapies, like, for example, checkpoint inhibitors or CAR T-cell therapies in the context of immunotherapy. These platforms can also be used with patient-derived cells, and then you can form these 3D structures to replicate the spatial and cellular diversities that you typically see in tumors, allowing them for more precise evaluation of various therapeutics, including immunotherapies or their combinations with chemotherapy and radiotherapy. This is specifically important because tumor itself is highly heterogeneous, and it's formed from diverse cell types with different genetic mutations, and then also spatial variations within the tumor microenvironment, which significantly impacts the efficacy and safety, and then also resistant mechanisms of therapies. There are also other immunological complexities that traditional models fail to mimic. For example, resistance to checkpoint inhibitors, immune-related adverse events, and also biomarker limitations are among those parameters that traditional models cannot mimic. However, organs on a chip systems can be designed in a way that they can test novel therapeutics with potential synergy to existing treatments, allowing for circumventing the issue of resistance mechanisms, such as, for example, checkpoint inhibitor upregulation, checkpoint program death ligand 1 or other types of protein upregulations. They also enable personalized treatment by using patients' own cells, including patients' own tumor cells or patients' own blood cells and T cells that can be injected into these microfluidic systems and then being used for testing testing different doses and combinations of drugs. And then also this platform, they can be used to validate newly diagnosed biomarkers like immune checkpoint expression levels using co-cultures of tumor and immune cells for improving the precision of immunotherapy. For these reasons, recently FDA, through their Modernization Act 2.0, they decided to put an end on obligations to use animal models only to test new drugs, which means that now organs-on-a-chip systems, if truly validated, can be used in conjugation with animal models for validation of drugs in preclinical testing. So the benefit of using organs-on-a-chip systems are the reduced costs, because when you use microfluidic systems, you're dealing with nanoscale volumes and small number of cells. And this way you can significantly reduce the cost associated with cells and then also with the reagent use. Of course, we minimize the number of animal use, which is aligned with 3R principles. Because these systems can be designed in a way that these assays can run in parallel. You can do faster iteration. You can include automation to improve reproducibility, and then by using AI, then you can analyze large quantities of high content data, and then that also improves the drug development process, and then identifying targets that can be eventually used for clinical trials. The research I'm doing in my lab is primarily focused on glioblastoma, which is a deadly type of cancer. This disease, the standard of care, which is basically chemotherapy, surgery followed by chemotherapy and radiotherapy, has not changed for more than 2 decades. There's only one chemotherapy drug available for this disease, and the life expectancy is 15 months at most. The disease is, or this type of cancer is highly invasive. The cancer cells infiltrate into surrounding brain tissues for up to a few centimeters away from the border of the tumor. The tumor microenvironment is highly heterogeneous. The cells rapidly proliferate and then quickly become resistant to the only chemotherapy that is available, and then the tumor itself, it has different genetic mutations between patients, and then even over time, the genetic mutations of the cells changes as they are exposed to the treatment. In terms of treatment, the challenges are the blood-brain barrier, of course, which restrict the delivery of therapeutics in the context of immunotherapy, immune cells, or antibodies, for example, to the tumor site. The issue of tumor heterogeneity is that we need to deal with diverse tumor cell populations that can lead to different immune recognition and response. Therefore, a single treatment is not going to be effective, and then we need to think about a combination of different therapies, perhaps different modes of treatments, including radiotherapy, chemotherapy, and immunotherapy together. But the challenge is what dose is the best dose to be used, and what combination is the best to achieve synergism. There is resistance not only to chemotherapy, but also quickly they become the tumor environment becomes quickly immunosuppressive. It is a cold tumor, which means that it does not allow the cytotoxic immune cells to infiltrate into the tumor and then eradicate the cells. Because the tumor is highly diffusive, its spread prevents complete removal surgically, and then enables the cells to evade the immune treatment immunotherapy treatment, and then the conventional 2D cultures, of course, they fail to replicate the glioblastoma's immunosuppressive microenvironment, and therefore there is a pressing need to develop such models. But before we can develop this model, we need to think about the tumor microenvironment, which is specific to glioblastoma. We need to look at the blood-brain barrier, and you want to make sure that a functional blood-brain barrier is being properly integrated into the model. We talked about heterogeneity, and then this has to be taken into consideration. We talked about why, because these different mutations or antigen expression require diversity in terms of treatment strategies. In terms of tumor microenvironment, we not only need to think about the types of cells that have to be included into the tumor, but we also have to think about modeling extracellular metrics. Also, there are things related to hypoxia, which impacts the cell function, and then also consider vascular components, specifically a vasculature that is leaky in these types of tumors. Then there are mechanical, chemical, and fluidic signals that the cells experience in the tumor microenvironment because of various stiffnesses, interstitial pressure, and then also gradients of chemicals that they have to be considered. And anything that you design, it has to be scalable and reproducible. Therefore, whatever technology that is being developed, it has to be compatible with automation, perhaps robotic systems, and then they have to have a high throughput for analyzing multiple drug candidates. So our approach is to use a technology, we call it Tumoroid-on-a-Chip. If you read my papers and then look into the literature about what we've done, there is also a term called Tumoroid-on-a-Plate. This is basically they're the same. They have the same meaning. Tumoroid-on-a-Plate is basically an insert, something like this, that we place into typical well plates. They fit to different well plate sizes. This is a 6 well plate format, but we have 24, 48, and then also 96 well plate format available. And then I'll show you a video of a 96 well plate format later on. So the approach we're using is to use these inserts and create these little tumoroids using cell lines or patient-derived cells, and then implanting these tumoroids into microfluidic components, which could be either integrated into our insert, or it could be a device that is independent of our platform, and then the tumoroids have to be transferred to that microfluidic device. So this technology, which we first introduced in 2018 in a publication in Lab-on-a-Chip, Dr. Seyfouri, who is the co-inventor of this technology, was the lead author on that paper, relies on microvils, little microvils that are arranged radially, 3,600 of them in this 6 well plate format, and they're connected to a loading chamber which is located at the center of each of these inserts. So the wells have the size in the range of 100 to 700 micrometer, which enables us to create tumoroid within the same range with high consistency. So these microwells are connected to this loading chamber via inclined microchannels. This is a unique design will enable us to seed the cells in the loading chamber, and then the cells roll down and distribute between all of these microwells uniformly to achieve cell aggregates and eventually tumor spheroids, which we call them tumors. So it's a one-step seeding process, no need to remove the extra or excessive cells, because the body of these Tumoroid-on-a-Chip insert is made out of a hydrogel, then it can directly be embedded into paraffin for immunohistochemistry or histopathology analysis. And because the body of this insert is transparent, you can use regular microscopy, fluorescent microscopy, confocal microscopy, using both upright or inverted microscopes. And then you can use immunostaining and different imaging techniques to visualize the cells, specifically visualize the tumors, and then see how these tumors respond to different types of treatments. So a few case studies. So in this case, Maytham, a PhD student in my group, decided to apply this approach to investigate the impact of chemotaxis within the tumor microenvironment, and then investigate the impact of this chemotaxis on the tumor remodeling within the tumor microenvironment. So we first generated the tumoroids using the insert that I showed you from glioma cell lines, and then transferred these tumoroids from the inserts and place them into an open surface microfluidic device that you can see here. So this open surface microfluidic device has two main reservoirs, a zinc reservoir, and then also a nutrient reservoir. And then in the center, there is a chamber in which we place the tumoroids, and then embedded in an extracellular matrix in a chamber. And then when you add basically a protein or a molecule and then the nutrient in the source reservoir, then we were able to generate a gradient of this protein within the tumor chamber. And then we were able to investigate the impact of these gradients on the tumor remodeling. In this case, we used fetal bovine serum as for proof of principle to mimic cytokine growth or growth factors distribution, and then expose these glioma cells to these gradients. And we assessed how these gradients will impact the tumoroid shape and remodeling. So one thing that we observed was that the tumor definitely responded to this gradient in a concentration dependent manner, and then this demonstrated the fact that we can now use this platform to investigate the impact of chemotaxis on tumor remodeling, potentially for recruiting regulatory T cells and creating an immunosuppressive tumor microenvironment. In a separate study, we investigated the impact of matrix stiffness on tumor remodeling using this platform. Again, we followed the same approach, but to mimic the dynamic stiffness variations in the tumor microenvironment, we exposed these tumors to gradients of an enzyme that degraded the extracellular matrix within the microfluidic device. We think that this approach better replicates the glioblastoma microenvironment, where spatial matrix degradation drives invasion and remodeling. So our results yielded two key findings. As we expected, enzyme gradients increased tumor invasions in a concentration-dependent manner, but also we were able to recapitulate two well-known invasion patterns of finger type and ring type that are typically observed in clinics, suggesting that there are diverse remodeling mechanisms involved, and this is potentially due to the presence of different cell types and different cell phenotypes within the tumoroid model that we've developed, similar to what has been observed in clinical images. we also wanted to further accelerate the process of investigating how tumor grows and then invades. And then for that reason, we developed a theoretical framework that predicts tumor remodeling and invasion with the goal of optimizing the therapeutic strategies in silico before even going into in vitro studies. The model has two components. A continuum component that models the tumor mass and its growth. And then there is a discrete component that is coupled with the continuum component at the interface, and it models the invasive parts of the tumor. So the discrete model follows the cells individually and then predicts how fast they grow and what pattern they grow within the surrounding matrix. Also, one thing about this model is that it's modular, which means that you can add different cells and modules considering their crosstalks with the tumor cells. This way you can add different components, add to the complexity of the model by adding different components of the tumor microenvironment to the model, and then increase the complexity by incorporating cell-cell interactions and then signaling pathways to study their effects on tumor remodeling. For example, in this case, we looked into the impact of neurons on tumor growth and invasion in two cell populations, one temozolomide resistant populations and non-resistant cell lines. And then we validated the model with our Tumoroid-on-a-Chip platform to ensure accuracy. The validation showed that the data, as you can see here in both, the field symbols and then in different colors, all with neurons and without neurons for both temozolomide resistant and non-resistant cell populations, we achieved very good and remarkable accuracy in terms of modeling the increase in the tumor size or changes in the tumor size, and then also invasion patterns and then invasion length. But one thing that we observed here was that the presence of tumors observed that the impact of neurons was significant in terms of tumor growth in non-resistant cells compared to the resistant cells, while the invasion was not that impacted by the presence of neurons. And this will prove that the presence of neurons can be crucial, specifically when you want to investigate the tumor growth within the tumor microenvironment. And then also these neurons are critical for modulating immunotherapy response, and our model can be used to design this synergistic treatment to overcome the drug resistance by using different approaches. So we look deeper into now the proteomic profile of the tumoroids, both when they were made out of temozolomide resistant and then also non-resistant cells. And then we also compared them to 2D culture, to the cells that were grown in a 2D culture, traditional 2D culture systems. You can clearly see that there's a huge difference in the patterns, in the protein patterns of these different groups. Specifically, there is a significant difference between the proteomic profile of cells grown in 2D versus 3D, And then also there is a significant difference in the profile of non-resistant and resistant tumoroids. In fact, we identified 1,100 differentially upregulated proteins that corresponded to 3D culture and resistant characteristics. And specifically, we identified proteins associated with iron metabolism, including upregulation of ferritin light chain and ferritin heavy chain, which were upregulated 4-fold and then 7-folds. And that motivated us to look into the possibility of using or intervening iron metabolism to be able to address the challenge of temozolomide resistance. And then for that reason, we looked into two therapeutics. One is the defruxamine, and then the other one was defriprone. These are medications that help removing extra iron from the body in a condition that is called iron overload. And then this iron overload is typically happen in people with blood disorder like thalassemia. Who they get really frequent transfusions. So what the approach we used was to create these tumoroids from both cell lines, and then in this study, patient-derived cells using our now we call it the tumoroid-on-a-plate platform. So this tumoroid-on-a-plate platform, the reason we call it the tumor-on-a-plate is because now we combine this microfluidic components of the device with the tumoroid formation components of the device, and then put them all together into one insert. Now we can make both tumoroids and then also recapitulate the tumor microenvironment simultaneously in one single insert. So the insert in this design has 4 compartments, and in each compartment we have 6 tumoroids and connected to a media chamber in which you can inject therapeutics, and these therapeutics will then be diffused into this tumoroid compartment via microchannels, side channels, to mimic the diffusion from blood vessels into the tumor. So the benefit of using this new design is that not only we can create tumoroids and then investigate their size change, but we can also look at invasion of these tumors into the surrounding tissue, specifically if you include both the tumor cells and then also all the tumor microenvironment compartments, including healthy cells, for example. So in this case, we were looking for identifying concentrations or drug doses of a combination of temozolomide and these basically iron chelating agents, to see whether we can find any synergism between these two drugs. So we expose all these different cells, including resistant and non-resistant cells, to different concentrations of temozolomide, and then also, and then also the iron chelators. And indeed, we were able to identify conditions, a combination of temozolomide and iron chelator concentrations or doses that yielded synergism, which was very promising. And then later on, we looked into how these concentrations impacted cell invasion. These are invasive cells from the tumor mass that you can see on the top. And then also we looked into how these drugs can or combinations can impact the cells we isolated from tissues collected from patients, in one newly diagnosed patient, and then another one was a recurrent patient, a patient with recurrent glioblastoma, which had temozolomide resistance as well. So this clearly shows that this platform can be used for investigating combination therapy using cell lines, and then also using different patient-derived cells. So one other thing that we wanted to do was to see whether we can recapitulate the immune signaling pathways, and whether this platform can be used in the current condition and complexity for modeling immune signaling pathways in glioblastoma, whether it can be used as a predictive model for immunotherapy applications. What the results that I'm showing you lacks immune cells for now, so that the innate immune cells are not present. It's basically only tumor cells and neurons that are present in the tumor microenvironment. But even though our model did not have the immune component, both patient data, which you see on the right, and then the data that we collected using our tumoroids, both the proteomic and genomic data, as well as the secretome data, show the upregulation of components and pathways linked to interferons, cytokine responses, and an innate immune signaling that indicates that our model can capture the tumor neuron crosstalk deriving immunologically relevant stresses and then interferon pathways. All of them are central targets in GBM immunotherapy. And then the overlap between our patient resistance data and signature and our models, enrich interferon cytokine response pathways, suggests that our model can be used to mirror clinically observed resistance-associated immune signaling, which strengthens our, the suitability of our model as a preclinical platform to test immunotherapy strategies. We also, when we were analyzing the data, we looked at the some of the genes that were also upregulated in or enriched in basically in temozolomide resistant cells versus non-resistant ones, and then the iron genes related to iron metabolism caught our eyes, and we think that this iron metabolism can link strongly with immune evasion and then also macrophage polarization. Specifically tumors, like studies have shown that tumors with altered ferritin iron handling, they often derive M2-like macrophage polarization, which suppresses anti-tumor efficacy, and therefore there is a possibility to use a combination of chemoimmunotherapy, potentially using ferroptosis and inducers such as Avastin, for example, to work in combination with immunotherapy to potentially address the issue of resistance to immunotherapy and even chemotherapy. Because Avastin, the mechanism of action is different than temozolomide. It can be also be effective in combating temozolomide resistance in cancer cells. So for those reasons, we looked at the Avastin, and we wanted to investigate how it impacts the tumors with the goal of using them for immunotherapy. Because we wanted to use immunotherapy, we decided to incorporate immune cells as well. In this case, we just used macrophages or macrophage cell lines, a well-known THP1 cell line in combination with the glioblastoma cells, and then a third component which is out of the scope of this talk. And then we wanted to form this tumor spheroid. This is a triculture spheroid system, tumoroid system, and made in our Tumoroid-on-a-Plate platform. One thing we noticed that when we used THP1 cells alone, they barely formed any spheroids. But as soon as we mixed them at different ratios with our glioblastoma cell lines and then the third component, then they were able to form quickly the tumoroids, and then as we changed the ratio, we were able to achieve achieve different size uniformities, and then different also sizes of the tumoroids. So then we exposed these to temozolomide, to Avastin, and then a combination of both of those. And then we also made these tumoroids from cell lines, non-resistant, resistant cell line, and then the two patient-derived cells. And then one thing we observed was that temozolomide itself clearly shifts macrophages towards a more pro-inflammatory M1, phenotype, and reduce the immunosuppressive M2-like population, something that has been also reported in the literature, that chemotherapy and then also radiotherapy can generate a more pro-inflammatory environment. They could be beneficial for immunotherapy. Combination therapy of temozolomide and Avastin was beneficial, especially for basically the temozolomide resistant cell line. But when we looked at the patient-derived cells, we did not see benefit. So the results we observed was line dependent, which raised some questions, interesting biological questions that we are now working with the PhD student Mahdi in my lab to answer those questions. So the last part of my talk is focused on how we use this platform to investigate drug delivery systems, specifically drug delivery systems that are used to locally deliver therapeutic payloads to the site of the brain. So we've been working on developing these types of drug delivery systems for a while, focusing by capitalizing on a material that was developed in 2016, which was shear thinning and injectable. They could be easily injected through medical catheters or needles, so to be delivered to the site of the disease using minimally invasive approaches. We built on that, and then we added chemotherapies to those materials. And then by adjusting the composition of the material, we were able to achieve different release rates. Then we also spend a lot of time on creating implantable, flexible, and hydrogel-based drug delivery systems, which could be 3D printed to the size of the tumor and then implanted later on into the tumor cavity during surgery and post-tumor removal. These are degradable materials, and then they deliver their payloads for up to a few weeks, and then to even improve the delivery system, and then remove the need for transferring the 3D printed constructs from the device or 3D printing machine to the site of the surgery. We developed this handheld bioprinting device that directly prints therapeutic payloads into the site of the disease. This has a microfluidic print head that enables manipulating up to 4 different materials simultaneously and sequentially, which enables us to achieve highly sophisticated patterns at the site of within the tumor. and then the drugs, once they are delivered, they diffuse out, and then they reach the sites of the tumor where needed. And then we are also working on making these materials more cell-friendly for cell-derived applications. But all of these platforms, for us, they require a testing system before they can be even tried in animals, because testing them in animals is really expensive. These are materials that have to be injected. There are considerations related to the release profile of the materials, and therefore for rapid testing, we think that using this Tumoroid-on-a platform is a useful tool, and for that reason we use our 3D printer, handheld 3D printed, directly 3D printed the mesh, for example, into this media reservoir that I showed you before in the Tumoroid-on-a plate system. And then they release their payload, and then the cells or the tumors that were formed in the tumoroid section were exposed to different concentrations of drugs, for example, and in this case, a combination of different drugs. And then this way, we were able to identify combination of doses of two drugs, in this case, iron chelator and the temozolomide, that was most effective in removing or eradicating tumor cells and shrinking the size of the tumor. So the last part of the talk is dedicated to our efforts. This is the credit primarily goes to Dr. Seyfouri, who combined bioprinting with our Tumoroid-on-a plate platform to add automation. And so one of the challenges in using these wells is the fact that when you want to deliver the cells or the extracellular component, we need to use pipettes, and then pipetting itself, it's labor intensive, and then it also causes challenges related to reproducibility and then delivery of the same amount of materials. So what we did was that actually what he did was that to create this 3D printer, and then adopted the 3D printer to deliver materials, including hydrogels and cells, directly into these wells. And then this way, he was able to generate arrays of spheroids or tumoroids up to 1,100 arrays per plate, using this automated system, which enabled him to also deliver multiple cells to create multi-culture systems, and then also to model invasion. So with that, I'd like to end my talk by acknowledging the financial support I received from many different funding agencies who helped us to progress in this space. There has been a lot of people working on these, many students, trainees, and then also my daughter, who always joins for launch gatherings, and then also I work with many collaborators who help us in this multidisciplinary research to be able to address some of the grand challenges related to cancer and drug development. So with that, I also would like to thank the Creative Biolabs team for inviting me again, and then also the audience for listening to my talk, and then I'm happy to answer any questions that you may have.
Thank you, Dr. Akbari. That was a great talk. I've learned a lot, and I'm very enthusiastic about this. Let me quickly share my screen again, so I can.
So maybe I should.
Yeah, I think if you can stop the screen share.
Yes.
So yeah, our audience, please feel free to start typing the questions in the chat. We do already have a few of them, so I will start with those. Great. Okay. So the first was actually not a real question, but more of a comment, stating that your organs on chips is a great uniform platform for anti-cancer drug screening. I'm absolutely in agreement with that. And apparently, some labs also showed that the anti-cancer effects of drugs can be markedly different between the 2D and 3D cell culture and transplanted cancer cells or 3D organoids in nude mice. So that's a comment. I'm not sure if you want to respond.
Yes, yes. This is a great comment, and I agree with you, Yanbin, if I pronounce your name correctly. So what we think is that these platforms can be used or can be used to more accurately predict uh, the screening part of the drug development process, right? So before we even test the drugs in vivo in animals, they can be used to test different therapeutics. Specifically, if you want to try a combination of different drugs, they can be really useful. But eventually, they have to be validated in any data that we obtain. They have to be validated in a more complex model until there is a more complex human-based in vitro model is available. This is my personal opinion. And at this stage, anything that is being suggested by the 3D models, they have to be tested in animal models, including nude mice, of course. But we are thinking about optimizing or improving the efficiency of the screening part, which is primarily traditionally relied on 2D culture systems, which are not true representatives of 3D complex environments that we see in our body. I'm not sure if I answered your question.
Yeah, I think we will move on with the next question. So does the also from the same attendee, does the iron chelating anti-cancer candidate have any effects on hemoglobin, myoglobin, or cytochrome C, etc., that contain heme molecules?
They can. These are great questions, and then but to minimize the any undesired effect, our plan is to deliver them directly to the site of the disease. So we would like to avoid systemic delivery as much as possible, specifically for cancer, for glioblastoma therapy. But there could be impact, and then we need to use the local therapy to mitigate any of these side effects that they may have.
Great, thank you. There is another question. First of all, great presentation. In your chip system, do your tumor spheroids develop blood vessel like structures? And if so, do they show tight junctions?
We don't have blood vessels, and what I showed you. Amir is working on making or integrating blood vessels, and I'm pretty sure he has done a lot of progress on that front. Amir is now independently pursuing further development on this technology. He's collaborating with me, but he's working on. I'm sure that he's working on integrating blood vessels into this. But the data will not be available in the next few months.
Okay, thank you. And then I think we'll go to the last question, considering we're running out of time. So last question, how does your organs on chip and tumor platform maintain patient-specific heterogeneity, and how reliably does this predict drug responses?
So this is a very good question, right? I mean the thing is, like, we're whatever that we develop is, and it's actually a very important question, is that we need to validate it against in vivo data and then patient data. So what the approach that we are following is that we looked at the genomic, proteomic, and metabolomic profile of our platform. We also look at the we receive the cells, we look at their mutations, and then see what mutations are present in our cell that we received, and then we compare that with the original tissue that we obtained. However, there is always effect of the tissue culture on the heterogeneity that you typically observe. So we look at the endpoints eventually, and then we test drugs, and then for validation, compare those results with clinical data. Again, this is a collaborating work with clinicians for further validation. It needs a lot of back and forth validations between our model and clinical model or clinical data. But that's definitely a valid point, and I think more work needs to be done to address this. But keep in mind that we are trying to improve traditional 2D culture systems, right? 2D culture systems, clearly they are not accurate. 95% of the drugs, they reach clinical, 90 to 95%, they reach clinical trial, they fail. So even if you can reduce that failure from 95% to 80%, still there's a huge gain, right? And then we're trying to address this great issue.
Thank you so much for for that insightful both presentation as well as answering of the questions. I think absolutely right. Indeed, the 3D model is trying to basically improve upon 2D. I'm sorry, there are more questions, but we're out of time, so I'm going to wrap up the seminar here. Again, thank you so much for this great presentation. Very exciting.
Absolutely.
And yeah, I think I'll leave the last slide to Kathy for concluding remarks.
Before we leave, Sarah and Kathy, is like, please feel free to email me if you have any questions about this platform. If you see any possibilities for collaborations, you can Google my name. Also my email is available, and then I'll be happy to discuss how any questions you have or how we can collaborate in the future.
Yeah,
Thank you very much again.
I think indeed there are great options for collaboration.
Yes.
Absolutely.
All right.
Thank you, Dr. Akabari, for your presenting. Yeah. Please note that the Creative Biolabs regularly invites estimated experts in life science and biotechnology to share their latest findings in our webinar series. So we encourage you to visit our website and subscribe to our social media channels so you won't miss any upcoming events. And that's it for today. Thank you everyone for joining us today, and we will see you next time.